Hello, Anusha.
Sure, do we have session tomorrow also?
Yeah, tomorrow also I will keep. Only Saturday, Sunday will be a holiday.
Okay, sir.
Yeah. Can you drop a message in the group for all to join?
Yes, yes, I texted in group.
Yeah.
So today it's only biostatistics.
Yes, only biostatistics.
Tomorrow supervised learning, sir.
Yes. No, tomorrow, yeah. Anusha, I'm transferring the host to you. Yeah, we'll start in another 2 or 3 minutes. Let us wait for some time for the others to join.
Yes, sir.
Yeah. Okay, we can start now. Good evening, everybody.
Good evening, sir. Is my full screen visible to all? Yes, sir.
Okay. Okay. So today let us start with the biostatistics. Tomorrow we will be taking the supervised learning, and Monday there will be image processing. So there will be no class on Sunday. Okay. So what is the biostatistics actually? If for analyzing the biological data, that is a separate term given for that is biostatistics. So there is no difference between basic statistics and biostatistics. This is just for the biological students, people who are studying life sciences or a biotech or bioinformatics and all. So they are given this term for called biostatistics. But everything is same as statistics, except for the part that the data sets which you will be taking will be of uh, biological origin. Okay. So it is just one short overview, because I hope that many of you will be knowing the statistics. So that first I will give a short introduction, then we will go ahead with the analysis, like how we can do biostatistics using Python. So father of biostatistics, Francis Galton, who was an English explorer, geographer, anthropologist, and statistician who laid the foundation of eugenics and modern statistical methods that led him to be known as the father of biostatistics. Okay, so the definition is crystal clear to all, I think, or like you can take any particular definition. So it is the branch of statistics that applies statistical method to the biological, medical, and health related research. That's all. So whatever is statistics, in that you need to add bio to that. Means it should be related with the for the bio students, medical, health related. Actually, for the even in the like clinical research or for this one public health, medical, they are using much of biostatistics, including the bioinformatics students. It involves the design, analysis, and interpretation of the data from the experiments and studies in these fields. An interdisciplinary field that helps that develops the methods and software tools for the understanding of the biological data. Biostatistics is essential for making the sense of the complex data, ensuring the validity and reliability of the research findings, and translating data into actionable insights in the health and biological sciences. So that is designing experiments, analyzing biological data, and interpreting the results. Okay. So for the data collection methods, already like we are using the experimental studies, that is the control experiments, the clinical trials. For then we are using the observational study, that is the cohort studies, case control studies, and cross-sectional studies and surveys and questionnaires. So these are the sample data which we are using. So key methods in the for bioinformatics, like mainly we will be doing the mean, median, mode, standard deviation, hypothesis testing, confidence intervals, PCA clustering, and so on. Okay, so today we'll be we will try to complete this mean, median, mode, then range, variation, variance, standard deviation, etc. Further, we'll be seeing the plots also, that is the histogram, bar charts, box plots. So this is a short definition. I hope that everybody will be knowing. Mean is the arithmetic average, median the middle value, the most frequently occurring the value in the data set. Then for the dispersions, we have range. That is the difference between the maximum and the minimum values. Variance, the average squared deviation of each data point from the mean. And standard deviation, the square root of the variance, providing a measure of the average distance from the mean. Then we have skewness and kurtosis. So I think you know that skewness is the measure of the asymmetry of the data distribution. We have positive and negative skewed. And for kurtosis, we have leptokurtic. Leptokurtic and mesokurtic. Okay, so other plots for your reference, histograms, box plot, then you have bar chart, scatter plots, heat maps, etc. Then for the interferential statistics, there is a branch of statistics that involves making inferences about a population based on the sample data drawn from the population. Underlying descriptive statistics, which focuses on the characteristics of the data set. Inferential statistics aims to draw conclusions and make predictions that extend beyond the immediate data. Key concept of the inferential statistic, that is, you have population and sample. So population is the complete set, the complete population, and sample is a subset of the population. Then you have parameter and numerical characteristics of the population, that is the population mean and population variance. Statistic and numerical characteristic of the sample, that is your sample mean and sample variance. And these are the common statistical tests we can use, like t-test, then ANOVA, chi-square regression, and so on. Then multivariate analysis. So in the multivariate, it is a very simple definition is that you will be using more than one data set. Very simple. So don't think that the multivariate is something very difficult. No, it is like there will be more than one data set. That is, it allows researchers to understand the relationship patterns, structures in complex data set where there are more than two variables. So two or more. That is what a multivariate analysis. Sometimes it will be like univariate, then bivariate also. In some books it will be there. Otherwise, it will be univariate and multivariate analysis. So you actually usually for the classes and all, we teach the univariate analysis, and later you can understand like how to perform analysis using the multivariate. Okay. So this is one of the very important analysis, and it is very simple. So types, you have multiple regression, then multiple analysis of variance, ANOVA, PCA, factor analysis, cluster analysis, discriminant analysis, and canonical correlation analysis. Applications in bioinformatics, there will be gene expression analysis in the diseases, protein structure and functional studies, medicines classifying the disease based on the patient characteristics and clinical data. In psychology, understanding the complex behavior traits by examining multiple psychological measures. In market research, you have segmenting markets based on the consumer preference and behaviors. And in environmental, analyzing climate data to understand patterns and predict the future changes. Okay, examples for your multivariate data. That is the age,
Sir,
income, that is
you
together.
explain about
Yes.
bioinformatics, the previous slide, the applications part.
What
The
happened?
first one in bioinformatics,
Yes.
the previous slide.
Huh.
Can you explain a bit more on that? The first one, bioinformatics.
Okay, in bioinformatics, you need to do the gene expression analysis. Gene expression, very simple is that from the gene, how you are getting the protein. That is in the transcriptomics, how you are getting the expressed genes and further the proteins. And further using the gene expression analysis, you can predict the statistics using the volcano plots. In protein structure prediction, you are using statistics for actually predicting the 3D structure of the protein. In modular software, you use Python, so and you are taking the statistical, you are taking the features of the protein which are already present in the database. Features are getting extracted in the unknown protein. So that is actually done with the help of statistics. So in all those areas in bioinformatics, the statistics is highly used. Actually, if you are using bioinformatics, actually the complete bioinformatics actually is relying on the statistics. Why? Because very simple, because bioinformatics analysis is based on the hypothesis. You are not getting directly. So in the wet lab, it is called as the actual data. In bioinformatics, this is the hypothetical data. So hypothetical, how you are doing it based on the statistics. You are doing SNP analysis, then you are doing expression, then you are doing exome analysis, then you are doing even this one, metagenomics. Everywhere it is statistics involved. Okay. So to perform multiple regression analysis to predict spending habits based on the age, income, and the educational level, you can use the PCA to reduce the dimensions of the dataset while retaining the most of the variability. Conduct a cluster analysis to group individuals with similar spending habits and demographics. It is essential for gaining the deeper understanding of the relationship within the data and making it informed decisions based on the comprehensive analysis. Okay. So in bioinformatics, you are using your statistics for the data analysis for the large data sets, that is for the genomic sequences, the protein structure, etc. Hypothesis for evaluating the biological hypothesis through rigorous statistical testing. Modeling, as I already told you, to build the 3D models of the proteins and the disease outcome. Visualization, creating clear and informative visual representation of the biological data, and ensuring validity, enhancing the reliability and reproducibility of the bioinformatics research by employing the robust statistical methods. So by integrating the biostatistics with bioinformatics, researchers can draw meaningful conclusions from data, leading to the discoveries in the area of genetics, genomics, and system biology. Then applications you are having in the genomic sequencing, gene expression analysis, and protein structure predictions. The commonly used softwares are your R, SAS, SPSS, Stata, excluding the Python, and the R is one of the most important software which I had even told in the last class that can be used, that is highly used for this one, statistics. And you have a bioconductor. Don't confuse, bioconductor is a R package. In R you have a CRAN and bioconductor. CRAN contains the general what you call packages, but bioconductor you have mainly the for the bioinformatics. Okay, then the visualization, you have ggplot2, MATLAB plot. Okay. Okay, so let us start with the statistics. You can open Colab and let us go ahead with the basic statistics.
Sir, your screen is not visible.
Yeah, one moment. Please open Colab. Everybody, you can open Colab. So my screen is visible to all. My screen is visible to all.
Yes, sir.
Yes, sir.
Yes, sir, it's visual.
Yeah. Okay, this code I am putting even in the chat box. So in Python, you need to import the module statistics and you can perform the mean. Mean, as you know, it is like it will be average. In R, there is no need to import anything. It can do directly. Just the command is mean. But in Python, you need to import the library and you need to execute it. So let us see if it is working or not. Okay, so it has worked. So mean you have got is 4.428. Okay. In the similar manner, you can even perform your median. So you can take this same data set.
Hi, sir. Sir, shall you explain once again, sir? Just now I have joined, sir.
Sorry?
Just now I have joined, sir. Can you explain this program once again, sir?
This program is very simple.
Means
It is
what are the
very-
concepts, sir, we're discussing now?
Concept is average.
Okay, sir.
Okay, because mean is average. So in this particular program, we are importing the module statistics and we are just applying that. You can see that x equal to statistics mean data 1. So that is, it is a one line code actually. This is the everything is here.
Okay, it will give the mean value for the given data one cassettes. The
Yes,
given values.
yes, yeah, that's all. Only thing is that you have to import this. In R,
Okay.
you don't even need to import, but in Python, you just need to import the statistics, and in that you will be getting this mean statistics. This is the function for this statistics.
Okay, sir. Okay, sir.
For median also, first thing what I will do is I will take this import statistics. Anyway, it is in the same code, it is not needed, but just for your understanding, I am taking it. Remember this, it is in the same notepad. So it is, this import statistics is nowhere needed again, but I am just showing it for your reference. Remember it. For the median, this is for the mean. Here it is a Python program to demonstrate the mean. This is Python program to demonstrate median. This is a median. So let us see if this is working. So median of the set is 4. How do you think it is 4? Actually, I have not explained the statistics. I hope that since it is a Python class, I am not taking the basic statistics, and I expect that you all know it. If you don't know, please let me know. What is median? Are you aware of this? Because there is only one command here. See here, for median, what I did was statistics median. Okay. So statistics median. Here I have used statistics mean. Here I have used statistics median. Now you have to tell me that you know what is mean and you know what is median. Is it fine for everybody? Are you aware of this?
Sir, no, sir.
You are not aware of basic statistics.
Sir, so long back, sir, I didn't actually remember, sir.
Okay. Because this is hardly a weak class. It is difficult to explain all these things. Okay,
Sir,
no
just
problem. I will
explain the
just
program of
I
how
will
this works, sir.
just
That's fine, sir.
I will just tell you. I hope that because as I told in bioinformatics, at least you should know the very basics. Otherwise,
Sir,
it
I
becomes
will learn
difficult.
myself, sir. Not an issue, sir. You can continue the class, sir.
Just I will tell anyway. Mean is the average. Mean is the average. I hope my screen is visible.
Yes, sir.
And then median is the middle value. So example like so you have your 1, 2, 3, 4, 5. Here it is understood that the middle value is 3. Okay. Here the middle value of this is 3. But if you are taking a set like this, one, two, three, 4, 5, 6. So in this, the middle value will be the average of these two. So that will be 3 plus 4 divided by 2. So that is 3 plus 4 is 7. 7 divided by 2 is 3.5. Okay, here the median is m is equal to 3. Here it is 3.5. Why it is 3.5? It is the average between 3 and 4. And then you have mode. That is the highest occurring frequency. Okay. So I hope you have typed it, or do you want me to put the, I will be putting this code in the chat box for your reference. Okay, so you can take it out, take it for yourself. Put it on chat box. It is only in the chat box only, no? No, it is not in the chat box. I put it in the chat box only. Okay.
Yes, sir. It is there.
What was the chat box? I could not understand. The message
Chat,
is
sir.
in,
Chat.
it
Instead
is
of
chat box only, no?
yes, yes.
Yeah, that's why I was saying, what is chat box? Okay. Okay, now same for the mode. So import statistics. So again, I am using the same command. Don't mistake here again that why I am every time giving, because it is the same notebook. Okay, but that is just for your understanding that we need to import statistics for even for performing mean, median, and mode. Okay. Now it is up to you to understand that what can be the mode.
It is 4.
Sorry.
4, sir.
Okay. Any others?
Yes, sir.
Anybody?
Same answer 4, sir.
Why it is 4?
A
You can
median
see
value,
here
no,
I
sir.
have
4
written
and 5, sir.
1 is 1 time, 2 is 2 time, 1 time, 3 is 2 times, 4 is 3 times. So we can interfere the 4 has the highest population distribution. So the mode is 4. Okay.
Okay, sir.
I hope it is clear with all.
Yes, sir.
So let me see what is the output, what it comes. Okay, so let us see what is the output. Yeah, mode is 4. Any doubts so far? Any doubts?
No, sir.
Okay. Now, before I move forward, anybody can tell what is standard deviation? Anybody? Anybody can tell you what is standard deviation? It is very simple. Is that if a particular data set you are taking, so what is how it is deviating from the mean? Okay, that is the simple definition for the standard deviation. Means how to what extent it is deviated. That is the standard deviation. So we have a formula. You can check it out in the this one. You can check out in the statistics book the standard deviation formula. Here it is very simple. That is statistics SDV. That's all, standard deviation. So even here, you are using, see, statistics. Import statistics, and the function is statistics stdv. Okay. So for everything you are using the statistics. Remember that. Even you can do using NumPy. There are various methods. Statistics is one of the most things that you can do maximum of the statistical functions using this statistics module. So let us see if at all you are getting this or not. So you are getting the standard deviation. Now we will do mean deviation from mean. Okay. So actually you can do mean deviation. You can do using median as well as mean. So it depends how you can, what is the requirement. I will just tell you how you can do that. Okay, did I put this code in the chat box? Let me check. Sorry, I did not put it in the chat box. Okay. Are you all doing it along with me? You can change the values and you can perform this codes. Again, you are using this one, we are using mean deviation from mean. This is okay. So here you are getting the mean.
তবে তোমার জুতো দিয়ে নিয়ে নিচ্ছি আসছে বাবা রাখ তাড়াতাড়ি রাখ
Okay, see if it is working or not. Here what I have done is I have given a particular list and then I have first here it is like mean you are getting it. So for each element in T, so one by one it is taking. So RES append function it is taking, and for the each and every value it is performing what is the deviation. So let us see if it is working or not. So it is working actually, mean deviations for each and every value it is working. Okay, you can take it. Did you all perform it? Is it done for all? Is it done for all? Okay. So we'll go ahead with the ANOVA. Anybody can tell what is ANOVA? Why we are doing it? What is ANOVA? So ANOVA, usually that is the abbreviation for analysis of variance. So for each and every item, suppose for a very simple example from biology I can give. So if at all you are for a particular media, if at all you are changing the temperature, so if at all you are changing the temperature, is the pressure getting changed or no? Sorry, if at all you are changing both, first case one, like if at all you are changing the temperature, is it altering the pressure of the media, and if there is any change in the media. So that is one condition. Second condition is that if you are taking two-way ANOVA, then if at all you are changing both the temperature and the pressure, what way it can affect the media. That is one more condition. Suppose now we can give one more example. Like you have, you are applying the same fertilizer to 5 different soil, and you are getting a productivity. So you can understand that based on the fertilizer, if it is the same fertilizer, if the productivity is altered or not. So if it is altered, means the soil variant will be changing. Okay, so in that is a one-way ANOVA, because the soil condition is getting changed. For the condition that the fertilizer given is the same. Now, if at all I tell that no, we are giving 5 different fertilizer to 5 different field, and the crop yield is the same, but not the crop yield. Crop is the same. So based on the yield, we can tell that yes, there is a significance between the soil and the fertilizer involved. So in this manner, we can utilize ANOVA for biological functions. Actually, ANOVA is one of the mean, median, and mode are very simple, but ANOVA is one of the most used function for any kind of biological data analysis. Even in agriculture, it is widely used. Agriculture, and even if at all you are performing the any wet lab experiment, if you put in a very simple wet lab experiment, just you can put this one and ANOVA statistics, and it is very easy. It becomes very easy to publish your paper. So just if at all anybody is from the wet lab background, or you can also tell to any person like biotechnology students who are in the wet lab to just to implement ANOVA, then the research paper becomes very much good, and it becomes apt for publications also. Okay, so we will perform the one-way ANOVA. So we need to install this SCI Pi. See if it is, first I have to see that if this is installed or not. If it is not installed, you need to install it. Yeah, so this is installed in Google Colab. That is why on Google Colab, many of the softwares are already installed. We need not to install this SCI Pi separately. Okay. So here production means, as I told just now, I gave the example for soil and fertilizer. So based on because here I am taking the one-way ANOVA. In this, I am giving this all our P1 is the production, like the when in a soil when you are applying the fertilizer. So what is the yield you are getting? So I have given there 4 outcomes. Okay, let us see what happens. I will just give this code to you. One moment. Let me know what is the answer you are getting. Perform it and let me know what is the answer you are getting. So you can see that it is very simple. And now you have to analyze the result. That is the one problem with we people, that just we are getting the result and we are keeping quiet. But you need to see already above we are told that mean, median, mode and all, it is fine. But here you need to analyze what result you are getting. Because statistics is not only about the results, it is not mathematics. So you now need to know, then let me know what is the what answer you are getting and what is the outcome. Anybody? Anybody can tell the analysis? Anybody? What is the answer you are getting? Here you are getting p value as 0.16. Okay, and statistics you are getting as 4.25. Usually, what is the p value range? It is less than 0.05. So it is less than 0.05, then you can tell that our results are significant. Okay. So this means that we can say that there is a difference in the production with respect to the fertilizer when it is applied to the soil, right? So we are not here telling the water, the fertilizer we are taking. It is just, you can imagine it is the fertilizer and it is the soil only, it is changing here, and you have various production. Okay, so in this manner you can actually use statistics in biotechnology or bioinformatics and so on. I hope I am clear with everybody.
Yes, sir.
I hope I am clear with everybody.
Yes, sir.
Okay. Okay, so maximum of the statistics usually needed for bioinformatics I have done for you. So tomorrow I will start with the what is that, supervised learning. Okay, so I will advise you to, if at all you are not with a statistics background, you can let me know so that if needed I can take a small class, because anyway it is difficult, but if at all you are not able to understand, please let me know about the things whatever I have told. I have told very basic statistics which is actually used in bioinformatics and related biological areas. So we usually mean, median, mode, standard deviation, and ANOVA is used. So you can practice the codes. Anyway, the codes are very, very simple. I have not given any difficult code for you. So you can check it out and let me know if at all you have any queries, doubts, you can always let me know.
Sure, sir.
So for today, I will be ending the class. Any doubts if you have, you can let me know. If no doubts, then I can conclude for today.
Thank you, sir.
Okay. Okay, then thank you so much. Good night.
Good night.
Good night.
Sir,
Yeah, I will call you. 1 minute. I will just disconnect and call back here.
okay,
One
sir.
moment.
Hmm.
It is good.